CADUCEUS is clearly exciting, a lot of things going on. You know, this morning, if you were paying attention to the cardiology panel, you understand that there's a lot of things going on with cell therapy, in cardiology in particular. The next company presenting is going to be Capricor. Dr. Linda Marban is coming up. I have to tell you that I was very surprised and excited when I first met Linda. This is one of the only companies that I've come across that made the transition from autologous to allogeneic, and I believe that Capricor really represents a second-generation cardiac therapy, because the best way to speak Japanese when you're in Japan is to speak Japanese. So maybe one of the best ways to repair a heart is to speak the language of what's happening at the cardiac cell level, and that's what Capricor is all about. Hi, Linda.
Thanks. Thank you very much. So most people speed past this slide, but before I do, I have to tell you that I got a call this morning from my legal counsel who said that they had sent an updated copy to my presentation, which assuredly means that I'm probably going to make some forward-looking statements, and my legal in LA are going to be eroding when they're watching this webcast. So thank you very much for your time today. I'm very excited to be here. This is an important conference for the field, and thanks to ARM, Jason, and Maxim, and Ted and Piper for allowing all of you to hear focused presentations on regenerative medicine, and also giving me the opportunity to share with you what we're doing at Capricor these days. We're a publicly traded biotech company focusing on developing novel therapeutics to prevent heart disease. We consider ourselves at this point in our development a pure play heart failure company. We were founded in 2005. Our first product was an autologous product. You'll hear more about that in a little bit. We were founded out of intellectual property from Johns Hopkins and the University of Rome. We then moved to Los Angeles in 2007, and our next major milestone was when we became Capricor Therapeutics through the merger with Nile Therapeutics in the most recent past in November of 2013. Capricor has built itself to a phase 2 clinical stage with probably the lowest equity investment, at least presented in this room, of just $12 million in equity investments, which has presented a very clean cap table for my investors. We've done that. We've built the program by accessing the work that's been done in our academic colleagues' labs, and also by having some grants and non-dilutive capital as well, which I'll detail on the next slide. We have several products in our pipeline. Today we'll be talking about our regenerative medicine product, the cardiosphere-derived cell, CAP-10002, or 1002. We also have some peptides in development for heart failure. We've decided to take a little bit of a different approach in building our company, going after the same type of therapeutic indication, heart disease, with different strategies. And we still have some things in our regenerative medicine pipeline. We're not going to talk about those today, but look for it in the next months and years to come. We have good intellectual property coverage from our academic licenses as well as from our internal program. Just to highlight, we've brought in $40 million approximately, just slightly under, in non-dilutive capital over the past few years, which has funded our R&D programs and our clinical trials to date. They're detailed here on this slide for you. The most notable is $19.8 million from the California Institute of Regenerative Medicine. We're a for-profit entity, so that's a loan award, but it's a loan award with very favorable terms for development. It doesn't have to be repaid unless the product becomes a commercial product and it's repaid on revenue. So that's very beneficial for us as we are able to kickstart our clinical programs without having to have raised money in the private or public markets. The two product candidates that we have in development are CAP-1002 and Cinderatide. CAP-1002 is our lead product. It's an allogeneic, off-the-shelf, cardiosphere-derived cell. It is being used now in the ALLSTAR trial, phase 1 and 2. Our phase 1 is complete, and you'll hear the data today for the first time on the safety of the cells. And we're currently enrolling patients in the phase 2 trial. As many of you may know, we entered into a collaboration and exclusive license option with Janssen late in 2013, and that relationship is well underway and going very, very well. And finally, we have a new clinical trial that you'll hear about today called Dynamic. Just to briefly provide an overview of the Janssen deal, for those of you that are not so familiar with it, we received an upfront payment for the option of $12.5 million. We also are, in addition to that, collaborating on the development of cell manufacturing. So unfortunately, we have to submit our slides days in advance, but if I could have redone my presentation this afternoon, I probably would have, because I'm so excited to hear that the field is finally paying attention to the manufacturing of cells as important as the clinical development of cells. And that has been the foundation of the development of Capricor. We've always believed that it's not very good if you have good clinical data, but cells that you can't provide in a broad spectrum to patients that are in need. And Janssen answered a lot of our needs with their skills and experience in thinking about pre-commercial and commercial manufacturing. And I think we've answered some of theirs by having a cell type that they are very interested in helping to bring to market with us, should all go well. They have the option to exercise their license anytime up until 6 months after the receipt of the ALLSTAR phase 2 data. They've agreed to use the 6-month data as a benchmark, even though ALLSTAR is powered against a 1-year infarct size endpoint. You'll hear more about that in a moment or two. If they exercise their option, we receive additional bio dollars, which may total up to $325 million. So a deal that's nice for the company, and a double-digit royalty in an escalating basis to be paid out on sales of commercial products. I think this deal is important to us, obviously, for the cash infusion as a baby company, also for the downstream potential capital opportunity. But I think what it really did for us is it really provided validation of the underlying science and the development that we had put into bringing CAP 1002 to where it is today. And it also obviously helps to support the field in general. I think all of us are pleased to see such a big player come in and make an investment in this space. Shown here is our therapeutic pipeline. I'm going to walk you through each of these trials individually over the next few moments. CADUCEUS was our phase 1 proof of concept trial done by our academic colleagues at Cedars-Sinai Medical Center and Johns Hopkins University. That was using our autologous product, CAP 1001. It led to the movement towards an allogeneic product paradigm where we are now conducting ALLSTAR phase 1 is now complete, and we're participating in recruiting phase 2. Dynamic is our newest trial in patients with advanced heart failure and using a non-stop flow multi-vessel infusion, so a slightly different delivery method and a slightly different treatment paradigm in much sicker people. And we won't be talking about Senderitide today, but that clinical development plan is underway. CADUCEUS was our proof of concept trial, used the autologous CDCs, so that meant that patients had a biopsy after their heart attack and then were called back when they received, when their cells had grown to dose and they received the cells down a coronary artery. They were patients that had a reduction in their ejection fraction, which is the ability of the heart to meet the needs of the body. And there were 25 patients treated in an open label fashion, 17 treated with cells and 8 standard of care control in a 2-to-1 randomization. It was a standard interventional catheter delivery to the infarct-related artery, sponsored by Cedars and Johns Hopkins, and the results were published in 2012 in The Lancet. So if you're interested in the details of CADUCEUS, either let me know or look into the literature. What the study showed is that we saw a significant reduction in the scar size, that is the amount of damage that was done to the heart. And we also showed a concomitant and, in fact, greater increase in healthy heart muscle in those patients that were treated with the cells as compared to the standard of care controls. This was the first time there was evidence of therapeutic regeneration in patients with post-myocardial infarction, cardiac dysfunction, and led us to move the company forward at a much faster rate. I'd like to just spend 1 second talking about what the CADUCEUS data might mean from a public health perspective. Shown here is a events curve collected not by us, but by Ed Wu and his colleagues at Northwestern, where they just looked at patients and the size of their heart attack and their risk of developing downstream events. And what you can see here is that patients that had a large heart attack, those that had a heart attack that encompassed greater than 18.5% of their heart, had a very high risk of downstream events, whereas those that had small heart attacks had a much greater chance of going on to lead a normal, healthy life. If we look and superimpose the CADUCEUS data onto this potential events chart, what we know is that both groups, those with treated and control, both started with an infarct size of 24%. The ones that were treated with CDCs saw a reduction of their infarct size to 12.5%. If these findings hold up in future clinical trials, it suggests that patients treated with CDCs, Capricor's cardiosphere-derived cells, have the opportunity to move from a high-risk group, or chance of a downstream event being relatively likely, to a low-risk group, completely and totally, at least reversing their chances, if not ablating the opportunity. So we think this was really compelling data. We decided to move forward, and then we had to sort of take a step back and say what went well with CADUCEUS. The data went well. What we didn't really love was the autologous paradigm. We were frustrated with the fact that we had to wait 3 to 5 weeks to treat people. Sometimes we couldn't treat people if there was a problem with harvesting, if there was a manufacturing failure through our fault or through the inability of the tissue to grow good cells. It's very difficult to maintain quality assurance and quality control. And the downstream person that's affected is the patient. It's not a manufacturing team that gets a little bit of a wrist slap if something goes wrong. It's a human being who gets a phone call and says, you can't get your cells. So we decided then to move on to the allogeneic paradigm. The advantages of the allogeneic cells are that we can semi-automate explant creation, we can make large master cell banks, many thousands of doses, and it's now FDA approved for clinical use. So we start with a donor heart, we dice it up using our semi-automated methods, we collect and make a master cell bank. Ultimately, they're put in suspension culture and made cardiospheres, which lead to CDCs in a monolayer culture, and ultimately the product is delivered to the patient. Our phase 1/2 trial is ALLSTAR. It's a randomized, double-blind, placebo-controlled phase 2, and it had a phase 1 open-label dose escalation safety phase, which is now complete. It's 30 to 40 sites in the phase 2 with intracoronary delivery. This is a schematic of what the phase 2 trial design and the phase 1 trial design looks like. The phase 1 was 14 patients. We have 2 cohorts in this clinical trial. We're looking at patients that are CADUCEUS-like, 30 to 90 days post-MI, and those that are a little bit more chronic in their injury, those that are 90 days to 1 year post-MI. And we are then going to compare and see if there's any difference in when you attack the injury. They're independently reporting trials in the phase 2, so when one is done recruiting, we can analyze that data. We have essentially a 2 shots on goal approach, and we'll be able to better understand where the efficacy signal is strongest in our cells. So now I'm able to tell you that ALLSTAR phase 1 is finished. We met our primary safety endpoint. Not only did we not only did we reach the safety endpoint, but we actually had no events that led to any potential safety signal, which was very reassuring to us, as this was the first time an allogeneic cardiac-derived cell was put down a coronary artery. The National Institutes of Health, which funded the phase 1, their DSMB allowed us then to move to phase 2. So even though we were relieved that we didn't see any major events, clinical events, regarding the delivery of allogeneic cells, we are a very strong science foundation company and don't just want to take that and move on. So we did a lot of immunology work in our phase 1 patients, which has been really helpful in helping us decide how to move forward with the phase 2 and some of the other decisions we've needed to make. What we found is that we really didn't cause what would ever be considered a clinical immune response. Some patients have pre-existing, and DSA stands for donor-specific antibody. One resolved that had the pre-existing, and 3 persisted, but they came in with these donor-specific antibodies, so it wasn't something that they mounted a response to the cells. 4 subjects developed low levels of DSAs. 2 of them resolved, and when we made the slide, it said 2 pending follow-up, but in reality, we've gotten the data back, and both have completely resolved to their baseline levels of donor-specific antibodies. Again, no clinical sequelae seen, and then we also monitored clinical immune response, our cellular immune response using the ALLSTAR assay, and while some patients actually had pre-existing responses to T cells, none of the treated patients developed any more, so that was reassuring to us. Looks like I'm almost out of time. The primary safety endpoint was for ALLSTAR phase 2 is the same. Of course, we'll continue to monitor the safety, including the immunology profile, so that we can, at the end of this trial, know a lot more about how the cells work. And the primary efficacy endpoint is a 12-month relative improvement in infarct size treated compared to controls, and we're looking for a 15% difference between CAP-1002 and placebo. The dynamic trial is one that is being conducted by our colleagues at Cedars-Sinai Medical Center. It's a very exciting trial because it'll allow our cells first time to be seen and are treated in patients with advanced heart failure. These are patients that are very sick with low ejection fraction, class 3 or ambulatory class 4 heart failure, and they'll have had a recent hospitalization for heart failure. Because these hearts are very sick and we can't sort of localize the injury, we're going to deliver the cells down all the coronary arteries, or in at least 2 of 3, and then we're going to be using non-stop flow, which hopefully will provide for greater comfort for patients during the infusion process. And that trial has got IND approval and IRB approval at our single center and should be beginning to recruit patients within the next 6 months. So finally, I'll conclude my conversation with you today by telling you why I think Capricor's CDCs are well positioned for success. We have a unique mechanism of action with the reduction in infarct size, the amount of damage done to the heart, and heart regeneration in the CADUCEUS trial. We've moved to an allogeneic product, which allows for an easy-to-use, off-the-shelf product adoptable by our cardiologists all over the world. We use donor hearts that can provide a sustainable source of tissue from which CDCs can be derived, and we can produce many thousands of doses per heart with a targeted gross profit margin of over 90%, and probably will be higher than that as we bring down cost of goods with our manufacturing modifications. We have a strong developing IP portfolio with many patent families under development, and we have a great team, which I show here, and most notably I'd like to call attention and thank my board of directors who have been instrumental in helping to build the company to this point. And with that, I'll say thank you for your time, and I'll be in the breakout room if you have any further questions.